Cellular aging is not inevitable decay. It is a state defined by epigenetic markers. In 2006, Shinya Yamanaka identified 4 transcription factors capable of resetting this state. These factors, collectively known as OSKM, can rewind the biological clock of any somatic cell. The goal is not merely slowing senescence, but achieving true age reversal at the cellular level. This process fundamentally challenges the concept of fixed biological lifespan, moving it into the realm of programmable biology. The factors function by binding to specific regulatory regions, initiating a cascade that dismantles the established identity of the differentiated cell. This breakthrough provides the molecular toolkit necessary for radical life extension strategies central to transhumanist goals. The Yamanaka cocktail consists of octamer-binding transcription factor 4, sex-determining region YBOX2, cripple-like factor 4, and proto-oncogene c-Myc. OCT4 and SOX2 are the core pluripotency factors, essential for maintaining the stem cell state. KLF4 assists in proliferation and survival during the reprogramming process. c-Myc, a potent oncogene, enhances the efficiency of reprogramming by promoting chromatin accessibility and rapid cell division. While c-Myc accelerates the process, its inclusion also introduces significant oncogenic risk, necessitating careful control in therapeutic applications. The precise stoichiometry and timing of expression for these 4 factors dictate the success and safety profile of the reprogramming attempt. Epigenetic reprogramming primarily targets DNA methylation, a key mechanism for gene silencing and cellular identity maintenance. Differentiated cells exhibit high levels of methylation at pluripotency-associated gene promoters, effectively locking those genes off. Introduction of the Yamanaka factors triggers the active and passive demethylation of these specific promoter regions. Active demethylation involves enzymatic removal of methyl groups, while passive demethylation occurs through dilution during rapid cell division cycles. Successful reprogramming requires the near-complete erasure of the somatic methylation landscape and the establishment of a new pluripotent methylation pattern. This global reset is what allows the cell to shed its age and specialized function. Beyond DNA methylation, the Yamanaka factors induce massive chromatin remodeling. Chromatin structure determines whether transcription machinery can access the underlying DNA. In somatic cells, pluripotency genes are packaged into compact heterochromatin structures. OSKM factors recruit chromatin-modifying enzymes, such as histone acetyltransferases and deacetylases. These enzymes alter the histone tails, primarily through acetylation, leading to a more open, accessible euchromatin state. This structural change allows the transcription of genes previously silenced in the differentiated cell. The coordinated action of DNA methylation and chromatin opening is mandatory for the cell to transition into a state of induced pluripotency. Full reprogramming results in induced pluripotent stem cells, or iPSCs. These cells are functionally equivalent to embryonic stem cells, possessing unlimited self-renewal capacity and the ability to differentiate into any cell type in the body. The creation of iPSCs revolutionized regenerative medicine by providing an autologous source of replacement tissue, bypassing ethical concerns associated with embryonic cells. However, full reprogramming completely erases the cell's identity, making it unsuitable for direct in vivo rejuvenation therapies. Injecting fully reprogrammed cells into a living organism results in the formation of teratomas, disorganized tumors containing tissues from all 3 germ layers.
The key to safe in vivo age reversal lies in partial, transient reprogramming. This technique involves expressing the Yamanaka factors for a limited duration, typically 2 to 4 days, insufficient to achieve full pluripotency. The goal is to reset the epigenetic age markers, such as the Horvath clock, without causing the cell to lose its specialized identity, for example, remaining a hepatocyte or a fibroblast. Partial reprogramming successfully rejuvenates the cell by improving mitochondrial function, reducing inflammatory markers, and restoring youthful gene expression profiles. This transient exposure avoids the catastrophic risk of teratoma formation, while still yielding significant functional benefits in aged tissues. Initial in vivo studies demonstrated the potential of partial reprogramming in mouse models of accelerated aging, such as progeria. Transient OSKM expression extended the lifespan of these mice by approximately 30%. More recently, partial reprogramming has shown success in restoring function in specific aged organs. For instance, studies have demonstrated the reversal of age-related vision loss by rejuvenating retinal ganglion cells. This targeted approach suggests that specific tissues can be selectively rejuvenated without systemic side effects. The challenge remains scaling these results to large mammals and ensuring the precise control necessary for human application. The primary safety concern in using Yamanaka factors is the inherent oncogenic potential, particularly due to the inclusion of c-Myc. Even partial reprogramming carries the risk of incomplete or aberrant reprogramming, potentially leading to malignant transformation. If cells linger too long in an intermediate state, they may acquire characteristics that predispose them to cancer. Furthermore, the formation of teratomas remains the definitive endpoint of uncontrolled full reprogramming. Future research focuses heavily on identifying safer factor combinations, often excluding c-Myc, or using small molecule cocktails that mimic the effects of the transcription factors, thereby reducing integration risks. Effective therapeutic delivery of Yamanaka factors requires precise control over timing and dosage. Integrating viral vectors such as retroviruses were used initially, but pose risks due to permanent genomic alteration. Current advanced methods utilize non-integrating systems, primarily modified messenger RNA or adeno-associated viruses, AAVs. AAV vectors are preferred for in vivo delivery because they can target specific tissues and provide transient expression, crucial for partial reprogramming protocols. Lipid nanoparticles are also being explored as a non-viral alternative, offering scalable and safer delivery, although targeting efficiency remains a technical hurdle. The ability to reset the epigenetic clock fundamentally shifts the paradigm from disease management to biological engineering. If cellular age can be repeatedly reversed, the concept of a fixed maximum lifespan becomes obsolete. Transhumanist goals envision periodic, systemic partial reprogramming treatments to maintain the body in a perpetually youthful state, achieving radical longevity. Future research focuses on identifying the minimal set of factors required for rejuvenation, potentially reducing the cocktail from 4 to 2 or 3 factors, minimizing risk. The next decade will see clinical trials testing transient reprogramming in age-related conditions, moving this technology from laboratory proof of concept to human therapeutic reality.